Hi, welcome to my channel. For centuries, we have understood biology as something we study. Today, we are beginning to understand it's something as something we can program. Cells are no longer just living entities, they are becoming computational systems. DNA is no longer just a molecule of inheritance, it is becoming code. And biology itself, it's becoming a programmable platform. So in the first part, I want to talk on the central dogma reimagined. So the first part is the central dogma. Is the central dogma. Well, this is just a brief explanation of the central dogma. The central dogma of molecular biology, which proposed by Francis Crick in 1957, and describes the unidirectional flow of um, genetic information. DNA makes RNA, and RNA makes protein, etc. Right. So classically, biology follows the central dogma: DNA, and then the RNA, and then the protein. But in programmable biology, this becomes code, and then execution and function. So DNA is source code, and RNA is compiled instructions, and proteins will be functional output. Right. Cells, in this sense, behave like input processing systems, signal interpreters, decision-making units. Biology is no longer descriptive. It is becoming engineering-driven. Now, in this part two, I want to emphasize on synthetic biology as a programming discipline. That would be part two. Synthetic biology introduces abstraction layers similar to software engineering. It contains of three parts. The first one is biological parts: promoters, enhancer, genes, equivalent to code modules. And the second part is circuits, with gene regulatory networks, equivalent to logic gates. And the third part is systems, engineered cells and organisms, equivalent to applications. And this has led to the emergence of three things: standardized biological libraries, reusable genetic components, design, build, test, learn cycles. And biology is now following the trajectory of computing in the 1960s. Now, in this part three, I want to talk about gene circuits, logic inside living cells. Right. So now, cells, they can be engineered to perform logical operations, such as AND gates, OR gates, and NOT gates. For instance, a cell can be programmed to detect cancer markers, yeah, which is input A, and detect inflammation signals, which is input B, and trigger drug release only if A and B are present. this transforms cells into autonomous decision-making systems, which is living therapies. Now, the next part 4, I want to talk on CRISPR as a programming language. So what is this? So the emergence of CRISPR-Cas systems, yeah, CRISPR-Cas systems has revolutionized biology. CRISPR is not just a tool, it is a biological programming interface. With it, we can edit genetic code with precision and turn genes on off epigenetic programming, inserts entirely new functions. Beyond editing, newer systems allow base editing, which is single letter changes, and prime editing, which is search and replace functionality. This is analogous to from low-level machine code to high-level programming languages, right? Now, the next part, which I want to touch on cellular operating system as in part 5. So if DNA is code, what runs it? The answer is a cellular environment, a kind of biological OS, well, which includes regulatory networks, feedback loops, signal transduction pathways. Cells interpret genetic instructions dynamically. So programming biology requires context awareness, environmental responsiveness, temporal control. So, and like software, biological programs are non-linear, stochastic, and adaptive. So they are non-linear, as in part 5. Part 5, touch cellular operating systems, right? Now, in this part 6, I want to talk on toward living software systems. We're now building systems where cells communicate with each other, populations coordinate behavior, biological networks self-organize, and examples include bacteria engineered to form patterns, cells that synchronize oscillations, microbial systems that compute collectively. This leads to distributed biological computing. Right. So now, how are we going to rewrite the living world with this in this part 7? And there are 4 areas that I want to touch on. The first one is medicines. Under medicines, we could use programmable immune cells, CAR T therapies, and smart drug delivery systems, disease detecting biosensor. And the second part is in the environment, bacteria that clean oil spills, carbon capturing microorganisms, and pollution detecting biosensors. Under agriculture, we could use crops that self-regulate stress, soil microbiome engineering. Well, under part 7, I want to emphasize this. Part 6 done. Okay, part 7, I want to emphasize this microbiome engineering. And For what is microbiome engineering? Well, this is a brief explanation for you. Microbiome engineering involves intentionally altering the composition, structure, or function of microbial communities to improve human health, environmental sustainability, or industrial processes. That is microbiome. And precision nutrient delivery. And as in another title, industry, under industry, there will be biomanufacturing, enzymes, materials, fuels, lab-grown meat, and biomaterials. And biology becomes a platform technology like computing. Now, in this part, I want um, to explain in part 8, debugging life, which is the challenge of complexity. So unlike software, biology is noisy, context-dependent, evolutionary. This introduces challenges, which is unexpected mutations, emergent behaviors, system instability. So debugging a biological system is not like fixing code. It requires iterative experimentation, probabilistic modeling, and systems level understanding. So it requires these 3 facts under here for 8. Yeah. Now, in this part, section 9, part 9, let's talk about biosecurity and ethical risk. Programmable biology introduces profound risk, like creation of synthetic pathogens, dual-use research concerns, genetic surveillance. And we must also ask, who controls biological programming tools? And how do we prevent misuse? And what governance frameworks are needed? This is not just a scientific issue, it is a global security challenge. Right? In this part 10, let me talk on evolutions might become design. For billions of years, evolution has been random, slow, selection-driven, and programmable biology introduces intentional design, rapid iteration, directed outcomes, and we are moving from natural selection to designed evolution. So under 10, we are moving from natural selection to designed evolution. Humans are becoming participants in evolution itself. Now, as with the convergence of AI, how do we include it in programmable biology? So it is in part 11. The convergence with AI. So the future of programmable biology lies in its convergence with AI. AI designs genetic circuits, machine learning predicts protein structures, automated labs execute experiments. This creates a new paradigm. And biodigital convergence, where biology becomes data becomes design, design becomes life. So that is biodigital convergence. If life can be programmed, modified, and designed, is life still natural? What defines an organism? Where is the boundary between machine and life? We are entering an era where life is not just discovered, it is created. So, as a closing of my public lecture, we begin by asking whether biology can be programmed. Now, we must ask something profound. What happens when life itself becomes a technology? When cells behave like machines? When DNA becomes code? When evolution becomes design? We're no longer just observers of life. We are becoming its architects. But with that power comes a responsibility to ensure that what we create does not outpace our wisdom. Because in the end, the question is not, can we program life? The question is, what kind of life should we choose to create? So this will be the end of my tonight's lecture for this video. And I hope you learned something out of from this video. And have a blessed night. God bless you bountifully in every area of your life. Bye-bye.